All right. Hi, everybody. My name is Sunny. Thank you, everyone, for allowing me the opportunity to present our study tonight at the webinar tonight. So the study that I'll be talking about, we started the study sort of early on in the pandemic with the first surge, and it's something that we're continuing on now with the more recent surges as well. So what we looked at was predictors of outcomes of COVID-19 infection in patients specifically with non-alcoholic fatty liver disease. We have no disclosures. So just to introduce our study briefly, so we've been dealing with COVID-19 for almost 2 years now, and really it's been consistently shown throughout the pandemic to disproportionately affect those patients with metabolic syndrome associated conditions. And there have been fewer smaller studies done throughout the pandemic that reported an increase of severe infection in patients with NAFLD. There's really not much more that we know in addition to this regarding the specific factors that portend adverse outcomes in this otherwise vulnerable group. And as many of us know, not only is the prevalence of NAFLD very high in the US, but it's also been rising over the recent years. And especially during the pandemic, this makes it even more important for us to identify any predictors of adverse outcomes in this otherwise fairly diverse group of patients. And this was the aim of our study. So just briefly about our study design, we performed a retrospective study, and this was done on patients with NAFLD that were diagnosed with COVID during the first surge at community medical centers in Fresno. And this is this is the largest tertiary care and safety net hospital that we have in Central California. And this was done during a 6-month study period between March and September 2020. Some of the information that we collected included baseline demographics, medications, lab values before and during COVID-19. And we also calculated baseline liver fibrosis scores as well. These included the NAFLD fibrosis score, the APRI score, the FIB-4 score, and the MELD sodium score. And we used all the variables to see if we could find any associations with certain measures of outcomes of severity, which I'll talk about in a bit. So a little bit about our statistical analysis. So we use chi-squared tests, independent sample t-tests, and Pearson correlation testing to assess for any correlations and associations. So we ended up including 298 patients in our study, and they're all over the age of 18, and they all had a diagnosis of NAFLD and PCR confirmed COVID-19. So now jumping into the demographics of our population, our mean age out of our 298 patients was about 55 years old, and it was fairly evenly split between males and females. Now, looking at the ethnicity of this group, you'll see that a pretty large percentage, over 70% of our population, is actually Hispanic. Now, we do serve a pretty large portion of Hispanic patients here in the Central Valley, and it's actually our most recent census data shows it's just under 50%. So we did see a relatively increased rate of COVID-19 infection among this group of Hispanic patients. And about 71% of our patients had a government insurance, so that sort of reflects the social status of these patients. Now, looking at the comorbidities, they're all the fairly typical comorbidities that we can see in patients with the metabolic syndrome or NAFLD, and most of our patients were obese, had hypertension, diabetes, hyperlipidemia. And one thing I'll mention on our table here is that most of our patients actually, in regards to the severity of their liver disease, actually did not have cirrhosis. Only 4% of them did. So over 95% of our patients were not cirrhotic. And one of the other areas of debate and sort of controversy in the literature now has been the association with medications and the outcomes of severity of COVID-19. So this is one of the things that we looked at as well. Specifically, we'll talk about ACE inhibitors and PPIs in our study. Now, looking at the outcomes of this group, it was a fairly sick group. About 83% of these patients ended up getting hospitalized. 75% of them needed supplemental oxygen during their stay, and about a third of them required more aggressive forms of oxygen in the form of non-invasive positive pressure ventilation or BiPAP. And about a quarter of them ended up getting admitted to the ICU, and we had a 14% mortality in our patients with a mean length of hospitalization of about 11 days. Now, looking at some of the lab values that we collected, we found that these were the peak lab values during their hospitalization. So we found that the ALT, AST, and total bilirubin, they all had statistically significant positive correlations with the outcomes of intubation, ICU admission, and death. And so essentially the degree of elevation of these labs was associated with these outcomes. And we found that the albumin and platelets actually had a statistically significant negative correlation with all of these outcomes as well. And albumin had the strongest correlation out of all of these variables. Now, one of the other things that we looked at was the association of medications, like I mentioned, and this has been sort of controversial in the literature in the general population, but in our study, we looked specifically at the NAFLD population. And what we found was proton pump inhibitors were associated with intubation and ICU admission in our population, and ACE inhibitors were associated with death, and these are all statistically significant. Now, one of the other areas moving forward, we also looked at the non-invasive fibrosis scores as well in this population. So the 4 scores that we looked at included the NAFLD fibrosis score, the APRI score, the FIB-4 score, and the MELD sodium score. Now I'll explain these graphs a bit. So if we look at the top left, we have the NAFLD fibrosis score, and along the x-axis, we have the measured outcomes of hospitalization, and they sort of get worse as you go to the right. So oxygen use, ICU admission, intubation, and death. Now, the orange line represents the scores in the patients, and these are baseline scores calculated on labs prior to admission and prior to diagnosis of COVID-19. So the orange line represents the scores in the patients who didn't have any of these adverse outcomes, and the blue line represents the scores in the patients who did have these outcomes. So as you can see, as the outcomes got worse, you can see there's a positive, even visually looking at it, that there's a positive correlation with increasing baseline liver fibrosis scores. And we noticed this trend with the NAFLD fibrosis score, the APRI score, and the FIB-4 score. The MELD sodium score didn't seem to show this correlation, and all of the MELD sodium scores seem to be fairly similar in the patients who had these outcomes and didn't have these outcomes. Some of the key takeaways from our study, based on our study, patients with NAFLD and elevated baseline liver fibrosis scores, elevated ALT, AST, total bilirubin, and decreased albumin and platelets during COVID-19 are at a significantly elevated risk for adverse outcomes. And when we look at baseline liver fibrosis scores, in our study, we found that the NAFLD fibrosis score, the APRI score, and the FIB-4 scores appeared superior to MELD sodium in predicting the outcomes. And we also, in our study, based on our study, we suggest that chronic PPI use and ACE inhibitor use were associated with adverse outcomes. And we suggest that they be used with caution in patients with NAFLD as we continue to treat these patients and navigate the pandemic. And these are all factors that we think that clinicians should be aware of when we're evaluating and treating our patients with COVID and NAFLD. So for acknowledgements, I'd like to just thank Dr. Reutman for all of her guidance during this project. She's really been a valuable mentor, obviously for fellows and as well as a lot of residents our program. So thank you, Dr. Reutman.
Thanks, Sunny. We'll leave the questions to the end. So if anyone has any, you can leave them in the chat box. So I'm just going to move on to the next presenter now, who is Dr. Aalam Sohal. I actually know him from when I rotated at UCSF Fresno. So he is from India, where he completed his medical education, and then he moved to Fresno, where he attended UCSF Fresno for his residency and is currently a third-year resident. Like Dr. Sandu, he's also interested in gastroenterology, hepatology, and is applying. And without further ado, go ahead and share your screen.
Thanks, Farah. Hi, my name is Aalam. I'm one of the residents here. The topic of my research was the pandemic within pandemic. So during the pandemic, we noticed that there was an increase in the hospitalizations for alcohol-related hepatitis. So that was the aim of our study. So we all know the third leading preventable cause of death is alcohol consumption, and COVID-19 led to significant anxiety, financial worry, and loneliness. We postulated that these factors were instrumental in the rise of alcohol sales and consumption. So our study was approved by IRB. It was done in the Fresno, California, the 3 major hospitals, and the data was collected from the electronic medical records. First of all, the subjects were identified with the EPIC SLICED assay. We used the ICD codes for alcohol-related hepatitis and alcoholic hepatic failure. About 420 potential subjects were identified through this. We excluded the pregnant subjects, and then we included the inclusion criteria. The inclusion criteria was alcohol misuse within 3 months of presentation, a white count greater than 10,000, AST-ALT ratio greater than 2:1, and elevated total bilirubin of greater than 1.2. From this, we included 329 subjects in our study. The data that we collected was all the patients that were admitted between January 1, 2019 to December 15, 2020. Demographics, basic labs. The labs were used to calculate the matrix discriminant factor and MELD sodium score. We looked at the interventions that were done on these patients, such as endoscopic procedure, intubation, ICU admission, and also studied the complications. So the results, first of all, coming to the demographics, the mean age between the patients of 2019 and 2020 was almost similar. There was an increase noted in all the age groups, but a major increase was noted in the age group of less than 40. There was 100% increase in patients with age less than 40. Also, there was an increase noted in the number of females. It almost like doubled as compared to the previous year. Although it did not reach statistical significance, it is still of concern. This is the percentage chart. We have like the percentages and the age groups. So we can notice that there was an increase of 8% and a 100% increase in patients under the age of 40. We attributed this to college campus closures, transitioning to online education, and lack of opportunities for socializing with peers. This associated with the stresses might have contributed to this increase among a younger population. We also noticed an increase in patients in women population. As we can notice, there was an increase in percentage from 18 to 27%, and 125% increase was noted in the women population. It has been documented previously that when exposed to a same psychological stress, women are more motivated to drink as compared to their male counterparts. We believe that this trend is of great concern as women have decreased alcohol dehydrogenase and they are at higher risk of developing alcoholic liver disease. This is a chart which explained the comorbidities. We had like similar number of patients between 2019 and 2020. None of that reached statistical significance. There were 31 patients with chronic liver disease in 2019 and 55 patients with chronic liver disease in 2020. Looking at the case numbers, as we know, between March and April was the time when the lockdown happened. And after that, we noticed a significant increase in patients in 2020 as compared to 2019. In December, there has been a decrease in the number, but we have to take it into account that we only collected data till December 15. It must have been higher if we had included the data for the whole of the year. Coming to the quarter-wise, in 2019, there were 31 patients admitted with alcoholic hepatitis. This number jumped to 69 in 2020 with a statistical value of P is 0.001. Same was noted in Q4, and the total increase was also statistically significant between 2019 and 2020. Why did this happen? We believe that many states had alcohol laws. Also within the early pandemic pipeline, we noticed a 54% increase in sales in the stores and 262% in online. We think that according to the survey that was done, 46% attributed this alcohol drinking to the increased stress. 34% said that there was increased availability of alcohol and 30% attributed to just being bored. And in that survey, 64% of the population said that it was a combination of all the above three reasons. We also calculated the score as well as the lab values. No statistically significant difference was noted between the Maddrey discriminant score, MELD score, albumin, INR, or creatinine between the two. Coming to the complications and interventions, we had a pretty equal kind of complications. The statistically significant decrease was noted in the endoscopies. There was a decrease from 41 in 2019 to 35 in 2020 with a statistical p-value of 0.03. And we also noticed an increase in the re-hospitalizations within 3 months. In 2019, there were 18 patients who were re-hospitalized, while in 2020, there were 35 patients who were hospitalized. We believe that the decrease in endoscopies was attributed more to the hospital COVID-19 infection control procedures, as well as the provider and staff being a little hesitant in doing endoscopic procedures in the COVID-19 positive patients, as well as in the setting of pandemic. The re-hospitalizations, we attributed that the patients did not have enough access to alcohol cessation counseling, and also the increased stress that was present during the time of COVID-19 pandemic. So discussion, the salient point of our study was that alcohol liver disease is on the rise in younger population as well as women. This burden was especially challenging during the times of COVID-19 as many hospitals were already beyond capacity. We noticed a 51% increase in the hospitalizations in 2020 as compared to 2019, and 69% was noted during a 3-quarter time frame between 2019 and 2020. There was a 94% increase in the rehospitalization rates between 2019 and 2020, suggestive of more severe disease, and as discussed previously, that patients did not have enough access to alcohol cessation counseling. The limitations of our study was the retrospective nature, the small sample size, and the limited geographical reach. In conclusion, we believe that our community is likely a reflection of the nationwide pandemic of the alcohol misuse disorder. We believe that this would have long-term impacts on the national health system, and urgent public health interventions need to be implemented to combat the rising misuse of alcohol. Thank you.
Thank you, Aalam. Our last presenter today is Dr. Alexa Wiengarden. She completed her MD and PhD at University of Minnesota in 2017. Her graduate thesis in the labs of Dr. Michael Sadowski and Dr. Alexander Koretz focused on mechanisms of fecal microbiota transplantation, so FMT, for Clostridioides difficile infection. She then entered Stanford Translational Investigator and a postdoctoral fellow in the lab of Dr. Michael Fischbach, where she studies how to engineer commensal bacteria to influence antigen-specific regulatory T-cell responses in inflammatory bowel disease. So you can guess what she will talk about today. Go ahead and take it away.
Awesome, thank you so much. All right. So yeah, as was hinted, I'll be talking today about some of the work I've been doing in the Fischbach laboratory since I started there several months ago, trying to understand some of the links between the gut microbiome and colonic T cells, specifically in inflammatory bowel disease. I have no disclosures. So as we all know, inflammatory bowel disease is an increasingly prevalent disorder, both in the developed world as well as in developing countries. While our treatments for inflammatory bowel disease have markedly improved over the past several decades, there's still a number of patients who kind of fall between the cracks in terms of either response to therapy or whether they lose response to therapy over time. So for example, for anti-TNF, it's estimated some 25 to 30% of patients are primary non-responders. And then another 30 to 40% of patients lose response over time. They become secondary non-responders. And so there's still a really large gap in terms of trying to effectively treat this disorder. As you guys are probably all very well aware, one of the major issues in inflammatory bowel disease is that although we call it a single entity, it's really not. Obviously, we typically divide patients into either ulcerative colitis or Crohn's disease, but even that is really an oversimplification. So within UC, for example, you can have patients who have just left-sided colitis or pancolitis. And similarly, in Crohn's disease, obviously there's multiple locations of disease along the GI tract, but even just within Crohn's colitis, you can have uncomplicated disease, you can have strictures, you can have penetrating disease or perianal disease. IBD is, as we all well know, is a highly heterogeneous disease, and that probably explains why we have such difficulty controlling the disease in so many patients, unfortunately. We also know, and this has become increasingly clear over the past decade or so, that the gut microbiome has a substantial role to play in inflammatory bowel disease, probably in both pathogenesis as well as in ongoing inflammation. So for example, if you take mouse models of IBD and you raise them under germ-free conditions, or you give them antibiotics to suppress the microbial burden in the gut, those models will often develop either less disease or no disease compared to their specific pathogen-free counterparts. We're increasingly learning that a lot of the role that the gut microbiome may play in IBD is within crosstalk between the gut microbiome and then both the epithelial cells that line the gut, as well as the underlying mucosal immune system. Normally, and you and me and everyone who doesn't have IBD, these different elements of the of this crosstalk are in harmony, and everything remains in balance. You have an intact epithelial barrier, and there's no inflammation. Or if there is inflammation, it's only appropriate, such as for control of a pathogen, for example. Unfortunately, as we well know in IBD, this is not the case. We don't know, for most patients, we don't know what the underlying trigger is, but ultimately most patients with IBD will have alterations in their gut microbiome alterations in the response of these immune cells that align the gut. And again, unfortunately, in these situations, patients will get inflammation. They'll get disruption of the gut epithelial barrier and all the symptoms that we unfortunately know so commonly happen in IBD. Okay. My goal with my research is to try to understand this crosstalk, and in particular, to try to understand how the gut microbiome can influence different populations of cells within the gut immune system. My work is particularly focused on different types of T cells that line the gut. So that's a type of lymphocyte. And then in particular, I also want to apply this, and what I'm going to talk about a little bit today is trying to apply that understanding to how that might affect why we have these different subtypes of IBD, and whether or not we can, there are differences among these different subtypes that could explain why patients don't respond all the same to different therapies. Okay. In particular, what I'm going to talk about today is the work that I've been doing on trying to understand how colonic T cells can recognize different microbes in the gut. Of course, we know that these microbes will make different metabolites. That's not the focus of this talk. And mostly I'm talking about different peptides that are produced by these microbes that are then recognized by different T cells, so a so-called antigen, a short peptide sequence from these bugs. The work I'll be talking about today is work that I've been doing in conjunction with another postdoc in the Fishback lab, Kazuki Nagashima, who really designed one of the assays I'll be talking about to work in mice, but we've been working to expand it in human patients. Okay, so what we've done so far, we've collected a number of different patient samples and have tried to identify different T cell phenotypes in the colons of these patients compared to healthy. What we've done so far, so we're focused on, I mentioned a couple of different T cell phenotypes. One of them are regulatory T cells, or Tregs. This is a population of anti-inflammatory T cells that can actually control inflammation that's caused by other T cell phenotypes. So we're interested in seeing what the differences might be in these populations in patients. This is pretty limited so far, but what you can see is that perhaps unexpectedly, given what we know about regulatory T cells, but not based on what's been previously published, there's actually an increase in the abundance of these regulatory T cells in Crohn's disease compared to a healthy individual. I'm just showing data right now from two individuals, and a patient with Crohn's disease, either an inflamed region of the colon or non-inflamed compared to a healthy individual. And this has actually been shown previously. So this fits with what we know about the different phenotypes of T cells in the gut in IBD. Another population of cells that we're interested in are so-called Th17 cells. This is a population of inflammatory cells that are thought to drive the pathogenesis of the disease, at least at some level, presumably in both UC and Crohn's, but obviously there's a lot of phenotypes of subtypes of IBD that we don't really understand the role of these cells necessarily very well in. What we've seen so far in this same patient is that the abundance of Th17 cells actually isn't all that different, at least in this individual compared to a healthy individual in the inflamed segments of their colon, but in the endoscopically non-involved segments, there actually seems to be an increased abundance of Th17 cells. And then, of course, I mentioned that the kind of the meat of the project is not just looking at different T cell phenotypes within the gut, but also trying to understand how specific microbes are recognized by different populations of T cells. And to do that, we're using an assay that I mentioned was developed by Kazuki to use in mice. And it relies on the fact that you have a very lock and key specific interaction between different T cells through their T cell receptor or TCR, and then antigens that are a short peptide sequence that can come from anywhere from the self, from food, or from gut microbes. And when these antigens are presented appropriately by an antigen presenting cell or APC, you can get a response in T cells. So what we're looking at essentially is if you chew up different bacteria by an antigen presenting cell and then pick out the antigens from each of those bacteria, show them to a T cell. If that T cell has a TCR that's recognizing those specific antigens, you're going to get proliferation of those T cells, as well as differentiation into different subtypes of T cells, whether that's those Treg cells, the regulatory T cells I mentioned earlier, Th17 cells, or other subtypes. So the assay that we'll be using, that again was designed by Kazuki for use in mice, is an in vitro assay. And what this does is we take individual bacteria, so monocultures that have been assembled, that we have available in the lab that we've collected either directly from human individuals or we've acquired from a biobank. We culture these with these antigen presenting cells, or culture. We heat kill the bacteria and then incubate those heat killed bacteria with these antigen presenting cells. And then we take those so-called loaded APCs and we culture them with colonic T cells that we've derived from patient samples. And the goal here, so these are, I mentioned that this can cause T cells to proliferate. Well, most of the T cells that are in the gut are cells that are, they're no longer naive. They've already seen their antigen and they've been told to get active and proliferate. However, if they see that antigen presented in the right way again, they will start to express this NIR77 transcription factor. And basically, we're using this as a way to tell whether or not the T cell that we're seeing in this assay is able to recognize the different bugs that we're giving to the different cells. And then we can also, not only can we see whether or not these cells are activated, we can also use flow cytometry to determine what type of T cells these are. Are these Tregs? Are these Th17, Th1, Th2, et cetera? I mentioned that Kazuki designed this for mice, and I'm very, very happy to say that we think, based on some of this early data, that we can use this assay in human colonic T cells as well. So what I'm showing you at the bottom on the left side is that same patient sample with Crohn's disease, and then the same healthy individual on the right. And on the bottom here, what I'm showing is the number of these NIR77 positive cells when the cells aren't stimulated. So we just took them out of the colon, we processed them, we stained them and ran them through the flow cytometry machine. But importantly, when we give them a non-specific stimulation, in this case, it's basically a chemical that automatically activates that TCR, you can see a substantial upregulation in NIR77 expression. So that means, and both the patient with IBD, the Crohn's patient, and the healthy individual. So that suggests that we should be able to use this assay in order to examine whether or not individual bacteria are also able to increase NIR77 expression in specific T cell populations, which would imply that those different T cell populations are recognizing those specific bacteria. So it's a really powerful assay in order to try to piece apart which T cell populations in the gut in humans, in both IBD and in healthy, are recognizing specific bacteria. In addition to that assay, we'll be doing some work that has been done similarly in other projects focused on IBD. So in particular, we'll be collecting not only cells from the gut, but also from the blood, both to run this assay and to do some basic flow cytometry like I had shown earlier. but also to do some single cell RNA sequencing and really try to dive into the very minute and specific immune phenotypes of these different cell populations across the different IBD subtypes. And then, of course, we're also collecting stool from these individuals predominantly so we can try to understand the different metagenomic content, the microbial populations that are present in these different subtypes of IBD, which again has not really been fully explored to the depth that I think is necessary to understand different IBD subtypes. And we'll also be performing some targeted metabolomics analyses, trying to understand what metabolites are produced by these different bacteria, which has already been shown to, various metabolites have already been shown to influence T cell identity and behavior, both in IBD and in healthy people. So that's going to be important to look at. All right, and then I mentioned we're mostly still in the sample collection process. So far, we've recruited just shy of 30 patients, both healthy and a number of different individuals with all the different subtypes of IBD, UC, and Crohn's colitis that I mentioned earlier. All right, so just to summarize, inflammatory bowel disease is, as we know, a heterogeneous disorder. But unfortunately, this complicates our understanding of both IBD pathogenesis and appropriate treatment. Understanding how different microbes are recognized in different subtypes of IBD might start to shed light on this. And then so far, what I've shown you based on my work to date is that colonic cell phenotypes do seem to differ between IBD and healthy individuals, some of which was known already. And then we're hoping that we can use this NIR77 assay to examine how human colonic T cells respond to TCR stimulation, and then shortly down the road, how these cells respond to the TCR stimulation that comes from an APC that's chewed up a very specific bug. So really how they respond to antigens from specific bacteria. Some of the future directions for this work, again, the key thing is moving on to testing specific bacterial strains in this NIR77 assay. And then of course, more deep characterization of T cell phenotypes in our patients, both using flow cytometry, basic flow cytometry to examine T cell phenotypes in the remainder of the patients we've already recruited. And then also performing single cell RNA sequencing, as I mentioned, to really get a deep sense of the immune phenotypes across our patients. And then, of course, characterizing the microbial populations in the patients, both by stool metagenomics, as I mentioned, and metabolomics as well. Okay, and then I just want to acknowledge, obviously, there's a lot of folks who have helped with this project and many, many other projects that I have ongoing in the lab and throughout the division. And of course, thank you all for attending.
Thank you, Alexa. That was very interesting. I had a quick question. I mean, this may be a little too early, but do you see your research being something that could be potentially targeted for treatment or prevention, or it's too soon?
That's a great question. So one of my other projects is more what I mentioned in my bio, is trying to engineer specific microbes to develop, really induce and or increase the populations of regulatory T cells to try to control inflammation. So this is sort of that's mouse work, right? But this is sort of the other arm where we're looking in actual human populations to see whether or not different T cell phenotypes are responding to different microbes. So for example, if we find that one particular Bacteroidetes species is really making the Tregs in one subset of Crohn's go completely bonkers, well, maybe that's a good species to target that we can engineer down the road to try to use as a potential biotherapeutic that's targeted towards that specific IBD subtype.
Cool, very cool. We had some questions in the chat. The first one I think or actually two of them for Sunny. So in your opinion, what is the mechanism for reasoning behind why PPIs would be associated with higher ICU intubation rate?
Sure. So yeah, great question. I think it's still unknown. I think, the literature there's a lot of theories. I think one of the most leading ones is, we do know that PPIs are associated with pneumonia, ventilator-associated vents, enteric infections. I think one of the things that the literature mentions a lot is when they looked at SARS-CoV-1, they found that there were decreased infectivity rates with lower pHs. And we do have a number of ACE receptors in the GI tract as well. So that might be one of the reasons that these patients are getting sicker and requiring intubation.
Great. The second question also for you is the population of patients with cirrhosis seemed low in your study. Is that reflective of your overall NAFLD population, or is there a higher proportion of patients with cirrhosis due to NASH who didn't develop COVID-19?
I think it's probably more so reflective of our overall NAFLD population. It's very high prevalence that we have. Very few of them end up developing cirrhosis. I think as far as the cirrhosis, I think once you do get cirrhosis, there's a lot of data that shows that the outcomes of COVID-19 across the board are worse in these patients. So I think these cirrhotics weren't necessarily, protected from their NASH, but I think it probably reflects the high prevalence that we have of NAFLD in our population here.
Just, I guess, to kind of piggyback, since we're picking on you, my COVID, or was it something else? And then do we know any vaccine status for them?
Sorry, Farah, you're just a little bit distorted there. I'm guessing that question was for me though, so do you mind repeating it?
Sorry. Yeah, so my question was whether the patients that you in your study, were they actually admitted due to something related to COVID, and were we aware of their vaccine status at that point? Maybe a little early.
Yeah, sure. No, great question. This the data that I presented actually was during the first surge. Most of these patients came in with symptoms of COVID. And 80, like I mentioned, 80%, almost 80% of them, ended up getting hospitalized for that. And a lot of them required oxygen. So most of these patients were related, the admissions were related to COVID. As far as the vaccine status, it was during the first surge, so most of them didn't. That is we're going to continue collecting data now with these recent surges, and that's one of the things that we're going to look at actually in this population. It's a vulnerable population, and are these patients coming in vaccinated? Or how many of them are vaccinated, and even things like which vaccines did they get is one of the things that we want to look at to see if there's any differences there.
Perfect. And then if anyone has questions, feel free to chime in. I'm just kind of asking. My other question for yours was whether these patients that were, if they were diagnosed histologically or by imaging of that they had NAFLD, or just based on kind of their comorbidities.
Yeah, so great question. I don't think I mentioned that in my presentation actually. So it was mostly due to imaging. Most of these patients didn't have liver biopsy or fibroscans on file. But they all had imaging on file. And obviously we ruled out other, alcoholic viral liver diseases and metabolic liver diseases as well.
Great. I had a question for Aalam. Which comorbidity did you find most impacted your conclusions?
So as I told in my presentation, there was not any comorbidity that was directly related. The only thing that made sense was that in 2019 there were 49 patients with a prior history of alcohol-related hepatitis, while in 2020 there was an increase to 78. That was the only comorbidity that was significant, but other than that, it was almost the same in both the years.
Cool, thank you. Let's see. I had some questions for Alexa, but I feel like it's kind of everything's theoretical. And so one of the questions that I had were, because you mentioned that nerve 77 is more with the gut integrity and the epithelial lining. Is there a reason why you guys chose to focus on that, or are there other genetic loci that are kind of being studied? And yeah.
Yeah, I'm sorry, I must not have been as clear as I'd hoped. So nerve 77 is not, it's nothing specific to the gut. It's a transcription factor that's found in all T cells that increases, so the cells start expressing it early on after they get a signal through their TCR. So the only way that they'll increase that is if someone is showing them, an APC is showing them an antigen in the appropriate way, or if we in the lab stimulate them, basically fake them out, like, hey, we're showing you an antigen, even though we're not really, it's just a chemical. But one of the key points of that is that unlike more traditional co-culture assays, where you grow the cells with those antigen presenting cells for a long time, it's a very short assay. It really only takes 4 hours or so for us to be able to see the increase in NER77 expression. So cells are they're more likely to survive, basically, because a lot of the time these cells can be pretty, you don't get a lot of them out of the gut, and they can die pretty quickly. So this is a way for us to be able to test how they're responding to different antigens, or antigens from different bugs that really hasn't been easily available before.
Okay, thank you. Does anyone have any other questions or comments? If not, I think we can probably end it there. Thank you guys all for attending and for the presenters. This was really interesting, and I hope we keep doing this. The next one is on February. If anyone has any comments, questions, you can always just email Danny, and then we can get back to you for that too. So thank you, everyone. Hope you guys have a good night.